Well, it's a pleasure to introduce Pam Small today. I'm going to take a couple minutes. Pam's journey has been very different than others, and I think for some of you, you might find it inspiring, if not interesting. Neither do I. Anyway, so Pam started out like most of us. Went to University of Wisconsin, a regular old standard good school. And then I'm not sure how it happened. Somehow she got married and had a bunch of 4 children and ended up in India with her family for 4 years, where she had a life-changing experience being exposed to the real disease. And I felt compelled by that to start volunteering first in India, and then wanted to know what makes people get sick. And I might say was perhaps naive, nevertheless, ended up landing at Stanford in Stanley Falco's lab, where she worked on diarrheal E. coli and found that the ability to resist acid in the stomach was essential for virulence. But still at that point, she wasn't ready to take on her own full-fledged research program. She went to Middlebury and had a bunch of undergrads who did pretty well and taught for 5 years or so, got tenure, then took a sabbatical at Rocky Mountain Labs and started making some significant discoveries in mycobacteria, and decided to move full-time to Rocky Mountain Labs, where she started working on the organism Mycobacterium ulcerans. Now, talk about neglected disease. How many of you even heard of it? As far as I know, Pam's the only one studying it in the United States. Nevertheless, you're going to find that Pam has taken this bug from the disease back to the lab, where she's discovered polyketide toxin that has amazing results, and now she's taking it back to the environment and trying to figure out the natural history, and it keeps going back and forth, and it's really interesting. Anyway, she went to University of Tennessee. She became a full professor. She stayed there for 8 years, semi-retired, but now is starting her own lab again at Rocky Mountain Labs in Montana. So as I said, it's a different path. Pam is one of these individuals in a field that is a kind of a glue that holds a field together. So everybody knows Pam, and Pam knows everybody. And she cares, and she has become one of the really best scientists in the field. And it's really, I'm proud to say that I've watched this all happen. Anyway, Pam, thanks for coming.
Thank you, Dan. Okay, well, quite an introduction. Thank you. I just never lived any one place too long my whole life. So I look at a map and I just want to go there. But what I want to talk to you about is an organism that's a very interesting organism. And it's caused by a mycobacteria, Mycobacterium ulcerans. Let me see if I can, which end of this works. Am I doing it wrong? Okay, I got the important things. And this is, as Dan said, a neglected tropical disease. And like many neglected tropical diseases, it's a disease that causes tremendous morbidity in areas where it occurs, but the distribution of the disease is limited. And it's limited probably by environmental factors, which we don't really understand. Now, about 10 years ago, World Health Organization started a program to study Buruli ulcer, to address the problem of Buruli ulcer. And this is the first map that they came out with. And every country that's red is a country that has had one case of Buruli ulcer. But the area where there really is significant burden of disease is here in West Africa. There's also disease in Australia. But the difference between West Africa and Australia is that people get early treatment in Australia, whereas in Africa, of course, they don't. So here is the disease, and when you look at this disease, it's really remarkable. When I first saw pictures of this disease in exotic Pathotica or something, I didn't believe it. And now, as I spend time in West Africa, I've seen hundreds of these cases. And if you just look at this lesion here, it's very remarkable for how clean it looks, and that's how they look. So it's a pusless, painless lesion. The organism only grows, causes cutaneous disease because it's temperature limited, like Mycobacterium leprae, so it really can't grow at body temperature. But what happens is, over the period of infection, these lesions get larger and larger and larger. If the infection occurs on a limb or an arm and actually 65% of the infections occur on the lower limbs. Very often you have a situation called auto-amputation, which is just what it sounds like, the leg falls off. So it's a result, it causes a lot of morbidity because now people can't work and people can't get married and people stop going to school. But interestingly, virtually no mortality, except for sometimes as a result of treatment. Now, there are a couple of very interesting things about this organism and its pathogenesis. It's a toxin-mediated disease, and I'll say more about this later, but this is a toxin, and if you look at this, it's a really simple molecule. It's carbon, hydrogen, and oxygen. You know, how can it do these things? You know, in fact, when I shared the first NMR structures of this, people told me nothing with that structure could have any of those effects. But this is the major toxin, and when you look at the pathology of the lesions, you get this sort of acellular necrosis in the center of the lesions, and it's called a coagulation necrosis. There's a lot of destruction of fat tissue. On the edges of the lesions, you see some slight inflammatory response. The organisms are extracellular. Well, any of you who study pathogenesis knows that TB is an intracellular bug, as are all other mycobacterial pathogens. This one is extracellular in the host. But when you treat the patients with antibiotics, which is something we've just started doing since 2005, as the bugs die, you begin to see the return of a normal granulomatous response. So the key to the pathology to this disease, or part of the key, is this toxin, which has a lot of immunomodulatory effects and also kills cells. Now, we have an animal model, a nice animal model, and after we had characterized the toxin and had found that it was a lipid, which made the characterization relatively easy, we were able to go back and identify the genes and look at this in an animal model. And really, you can recapitulate human disease quite well. This is in a guinea pig skin. And this is the wild type infection. You have some calcification here, but there's just sort of acellular necrosis in here. Very few inflammatory cells. We're looking at the dermis here. Here are the bacteria, huge clusters. It's almost like microcolonies growing in this lipid pool is the way it's been described. But if we knock out the toxin genes, now we get a lesion that really looks, from the standpoint of histopathology, a lot like TB. It's a granulomatous response, and most remarkably, here are all the bugs inside macrophages. Now, we actually explored the potential of this strain as a vaccine, and what we found was if you inject guinea pigs with this strain that doesn't make the toxin, you get no lesions, which wasn't surprising, but we could isolate the organism up to a year after infection, which is as long as we looked. So it still has the ability to survive in the host. And we're not doing any vaccine work. The problem with this molecule from an experimental standpoint, and it impacts everything, whether you're looking for the target for activity or trying to identify pathways, is that it's a lipid, it's not a protein, so you can't make antibodies to it. I mean, we have tried numerous times, but it's really hard to make an antibody to immunosuppressive lipid. So when we want to look and ask, is this molecule really in human tissue? The method that we've devised is to take human skin, do an extract from human skin, and use mass spectroscopy to see if we can find the molecule. And this is an epoxide of the molecule. That's the major toxin, and this is the core, and I'll say more about those later. So we can show that this molecule is present throughout the course of disease. We've just started this work, and so we'd like to look at this in a much more, in a more quantitative way. Now, I want to say something about the bacteria, because it's really an interesting bacteria. If you look at this chart, which is based on 16S RNA sequence, you can see that ulcerans and marinum are very close. And in fact, Mycobacterium marinum is a major infection in fish. And these two organisms differ by only one base pair in their 16S sequence. And they're actually very closely related to TB. In fact, one of the major things that they differ in is their growth temperature, which is a phenotype nobody really understands why both of these organisms can only grow at lower temperatures. They can't grow at 37 degrees. We had thought that ulcerans was sort of in a class by itself with respect to this toxin, and I'm going to say a little more about this, but in the last few years, we have found that there are several other very closely related organisms that also make this toxin. They have all been discovered in the context of environmental degradation. And the first one, I got the lead on this when I gave a seminar here 4 years ago. So I gave a seminar, and at that time, there was a group doing development biology. I've never met the guy who runs this, Harlan. His name is Harlan. And he had an undergraduate, Kirsten Trott, working in his lab, and there was a postdoc, Tim Grammer, and they wanted to meet with me. And they said, we've got this terrible mycobacterial infection in fish, and it has one gene that is also found in ulcerans, and we want you to look at it. When I looked at these fish, these frogs, these frogs were like balloons. They were totally edematous, and that is one of the symptoms of Mycobacterium ulcerans before you get the ulcer. So I took they gave me a plate of stuff. This was when travel was a little more permissive. I just brought it home, and really within like a few days, we were able to isolate a new mycolactome from this fish pathogen. And I'll show you the structures of these later. So this was very interesting because I had just had a graduate student do an entire survey of all known mycobacteria, environmental and pathogens, as well as uncharacterized mycobacteria from Africa, from the Congo. And the conclusion, which she published in a paper, was that there are no other mycobacteria, or that mycolactome production by mycobacteria must be very rare based on her data, which was a lot of work. And then we come here and we find this bug. So a year later, I'm reading about these new Mycobacterium marinum isolates that for the first time are causing disease in the Red Sea. There'd never been any mycobacterial infections in fish in the Red Sea, but they started doing fish farming. So this guy, Andrew Killarney, he sent me his strains of M. marinum, and they were very strange. They had some phenotypic differences, but again, they also are making mycolactone, a specific, slightly different molecule. And at the same time, an emerging disease in fish has been found in the Chesapeake Bay. And they had isolated a new mycobacteria from that, Mycobacteria pseudoschatzii, and it turns out that this strain also makes a toxin. So it turns out there's actually sort of a little family, a little cluster here of mycolactome producing mycobacteria. Okay, so with the help of WHO, as an advocate, we were able to get the genome sequenced. And I think there were two major findings from the genome. One was that the M. ulcerans genome is a genome undergoing reductive evolution. And you can see that because there are 727 pseudogenes. This is being driven partly by insertion sequence insertion. So there are over 100 copies of two insertion sequences, IS2404, IS2606. And when they land in genes, of course, they inactivate them. But there also are a lot of deletions. There's much reduced lipid biosynthesis. It's almost like these bugs are putting a great deal of their lipid biosynthesis into producing mycolactone, but they're also reduced ability to grow at low oxygen. So they can't use fumarate or nitrate as a terminal electron acceptor, which other mycobacteria can. So this whole genome picture is telling us something, but we really aren't able to interpret it. And the other very interesting finding was that the mycolactone genes are on a plasmid. Now, this is pretty exciting because we know that environmental mycobacteria, non-pathogens, do have plasmids, but no plasmid had ever been found in a virulent mycobacteria. And in terms of the plasmid biology, conjugation, and so on, I would say almost nothing is known about these. But now we have a cluster of species, all of which contain this plasmid, and the plasmids are highly homologous. We also know that in addition to these plasmid insertions, there have been these sets of deletion events that have led to this general evolution along the M. ulcerans pathway, and we think that the ancestral organism was this fish pathogen, Mycobacterium marinum. Now, this is the mycolactone chemical family, and these are all called mycolactone congeners. If you look from here up, there's something interesting going on here. There's a kind of geographic specificity to the form of the molecule. So if you see in Africa, all of the M. ulcerans have this structure. All the mycolactin molecules have this structure. This is the side chain, this is the core. The core, lactone core, is conserved in all mycolactones, but the side chain varies. So in Africa, you have this form, okay, and this is the most toxic. In Australia, you're lacking this hydroxyl at carbon 12 of the side chain. And when you get down to Lifflandii, the frog pathogen, and the fish pathogen, you lose a methyl group, and you also are lacking this hydroxyl. So what's interesting about this is when you look at the gene structure for these that encode the polyketide synthases that encode these molecules, they're highly repetitive. So they're in the lab, a lot of recombination goes on, and you get deletions very readily in different forms of the molecule. But in nature, we have looked at M. ulcerans isolates and extracted toxin from strains over a 40-year time period, and the structure is invariant. And this says to us that there's some kind of possessing a particular form of the toxin probably has some advantage in the particular environment, which is M. ulcerans' home. Now, I'm not going to talk about this, but I just want to put up a lot of references because a lot of work has been done on looking at the immunomodulatory effects of mycolactone. We know at high concentrations that are biologically relevant, it kills cells. But at concentrations too low to kill cells, and too low to even cause cytotoxicity, it inhibits various arms of the immune response and really seems to inhibit both innate immunity and to some extent adaptive immunity. A lot of work has gone into looking for the target for this molecule, and we spent a lot of time trying to do this, and I could give like about a week of seminars on our negative data. But basically, no, we haven't been able to come up with an identification of a target. We have good evidence there's not a cellular receptor, that it's like erythromycin or rapamycin. It enters a cell through a diffusion, but it's interacting with targets inside the cell. And until I heard, I think it was Elizabeth's talk, I would have said that DNA microarrays have led to more uninterpretable data than any other method. But maybe, I think we just don't know how to mine it appropriately. So we've done lots of arrays, but we're nowhere really with a target. And nobody else so far is either. Just quickly, diagnosis. Diagnosis can be done clinically when you have a big lesion. But this is a rural disease, okay? So it's occurring in small villages. It's one thing we know for sure in Africa. And so about the only method available in small clinics is acid-fast staining. It's not sensitive. IS2404 PCR is used in reference surfaces. This is one of the insertion sequences that seem to be unique to M. ulcerans. We know now it's not unique to M. ulcerans. So it's not very specific, and also it certainly can't be done in the field. And culture is very insensitive. Treatment, really until 2005, treatment was really crude. Just cut out the lesion and try to cut out as many of the bugs as possible, and then skin grafting. Now, if you have a kid like you saw in that picture, and if that kid's only 3 years old, you just can't find enough skin to cover that up. So, I mean, there is some mortality to the treatment, but really not to the disease, so far as we know. But in 2005, based on work by Jacques Rosset, doing animal studies of therapeutics, it seemed like using an aminoglycoside along with rifampicin, a typical TB drug, really had a lot of promise. And we have clinical trials, and it really looks extremely promising. Now, the problem with this is that they're using streptomycin, really cheap. There haven't been any problems with hearing or anything, but it's a 3-month treatment of an injectable drug. And they're very hesitant to introduce treatment at the local level in the villages. And I just came back from Ghana in October, where we're doing field studies, and basically the infrastructure is so bad that transportation is impossible, and you just can't expect people to go. They can't go daily to the health center. That means they have to disrupt their life and go for 3 months. So although this is proof of principle that antibiotics work, we need something better. Now, like I would like a company that could come up with a streptomycin patch, for instance, or an oral thing. And I've been told like it's a difficult problem because so far aminoglycosides and rifampicin seem to be the only thing that really works. So I want to end up talking about M. ulcerans as an environmental pathogen. And I'm not sure if, I have to ask John, I'm not sure if cholera is an environmental pathogen or not. It probably doesn't meet the right criteria. But in my way of thinking of this, environmental pathogens are pathogens for which, if humans disappeared from the face of the earth, the life of that organism would not be diminished in any way whatsoever. So people are accidentally infected. And sort of a corollary of that is that these organisms don't spread from person to person, or if they do so, very poorly. And what's kind of, so when I draw these two circles here, to sort of portray the idea, this is the life of the organism in the environment, this is the human, and they just intersect on a very narrow piece, sort of like a Venn diagram. And there has to be special conditions that bring these together to get disease. Now, if you look at this list of environmental pathogens, it's kind of remarkable. It's most of our select agents, right? And when you consider, you know, people often said, why are we spending all this money on organisms that only cause 100 cases of disease a year? I mean, and Stanley used to always say, well, I can't put it down because it's funding all my students. But the fact is the reason this is probably true is because there's not person-to-person transmission. And that's why it was really interesting to hear Julie talk about transmission, because if you were going to make a pathogen and you were going to enable it to spread from person to person, what genes would you add? I mean, it's a difficult question, a difficult thing to study. Here you have huge numbers of Legionella in the lungs, no secondary cases. And the same is true of ulcerans. All these organisms just packed, just almost like colonies of organisms in this tissue, and other people in the household don't get infected. So there's something here that we really don't understand, and I think it's an interesting question. So since it's an environmental organism, we thought what we'd like to understand is what is the natural environment for this organism? Where is it living? Where is it replicating in the environment? Can we identify the environment that promotes this organism in such a way to have an intervention? So for instance, we know with Rocky Mountain spotted fever and Rickettsia rickettsii, it's carried by ticks. The ticks are on the west side of the Bitterroot River. And if you're hiking, when the snow is melting, you don't want to be near the snow melt, because we know that's where the ticks are, and that's where you're likely to get infected with ticks. And if you get a fever, we'll know that it could be Rocky Mountain spotted fever. So we don't know very much about ulcerans. So we got involved in a disease ecology project funded by NSF and NIH with a group of ecologists from Michigan State University. And it's really enriched both of our groups, I think, a lot. And we're doing our work in collaboration with the Noguchi Institute of Medical Science in Ghana, part of University of Ghana, the Public Health Department in Benin, and also in Cameroon. So we aim to try to understand what are involved in disease emergence. It looks like this disease is occurring more often in people and that the distribution of the disease is changing. What are the factors driving that? How do people get infected? Even if you find it in the environment, how can you really show what particular contacts are important for infection? And then eventually, how can you develop intervention strategies? So to answer these questions, we have to consider a lot of data. It's very unlike the way we're used to thinking, at least I'm used to thinking. We have to collect data on everything. I mean, and that's pretty much what the ecologists tell me. And they think in a very different way. And what I've been told, I mean, which was really disturbing when we started this project, was if we sample everything in the environment, that's what we should do. And the important associations will fall out. So I'm always wanting, well, let's do 5 non-endemic, 5 endemic villages. No, we have to do 80 endemic, 80 non-endemic. They would like to do every village in the country. But I've repeated this. I've gone now to several ecology of disease meetings, and I've repeated this to other people just to see if this was just my group that felt this way. But it seems to be the general, they thought that was great that I understood that. So and it's not just that you're sampling all these different sites. I mean, you've got to gather all this data on everything. I mean, that really is the idea. And when you get all this data assembled together, you're going to express it, and this is where the fun part comes. So the field work is pretty straightforward. You're mucking around in water, collecting plants, insects, blah, blah. Then you come back and you have a mathematical model. Well, now this is a problem too, because most of us don't understand what they're talking about. And at these meetings, they put up these formulas, they don't define their terms, and they argue fiercely about them. And so it's a learning curve, you know, but the idea is to put all this stuff together in a big model. Dan asked me if I really believe in this. It's too soon to say. They've been very successful at taking data about diseases that are already known and modeling, showing that they can create a model that predicts what happened in the past, okay? And they don't like to do anything that incorporates new data too much, so we're sort of in new territory here. So these are some of our study sites, and I want to say something here that the red things, crosses, show you where Buruli ulcer, the disease, is present. The blue ones show where it's negative, and I think it's pretty clear to see that there's focal distribution here. So you've got this cluster here and this cluster here where you have Buruli ulcer, and yet you also have no Buruli ulcer in this village. Each dot is a village, but you have Buruli ulcer here. And then you've got this big region where there's no Buruli ulcer that's ever been reported in 100 years, and nobody had ever knew anything about it at all. What we know from the literature and from really around the world is that there's some association with slow-moving water. So it has something to do with exposure to slow-moving water. But I showed you that area where there were all those blue dots. That's the Volta Lake, which is the biggest water body in Ghana. No Buruli ulcer there. Seems to be associated with slow, small-moving bodies of water. And when you go out, in the past what's been done is people have gone out to the environment and they've looked. They'll pick up a piece of a lily pod, and they'll pick up 5 snails that they saw on the ground and 2 of this and so on. Go back, look by DNA for a signature of M. ulcerans. And doing that, it's been reported in many aquatic taxa, but not in terrestrial ones. I think the water connection is holding true. So in our study design, we decided that we would go out and look at the environment. Now, I should say something about isolation from the environment of the organisms. The organism grows on media, but it takes 5 weeks to form a colony, so it gets overgrown, you know, by everything else. But Françoise Portals in Belgium this year isolated the organism from a Gerardi. A Gerardi is a water strider. It is incapable of biting people. And she did this by collecting Gerardi, grinding them up, and passaging them through mice for 2 years. And this is the only environmental isolate we have, but it was proof of principle that the organism, it's not just the DNA signature we're seeing that the bug is actually there. These are some of the sites in Ghana associated with infection. This is gold mining. These are sort of little individual gold mines. And the water's near the surface, and when they dig these pits, they get filled up by water. This is the Ashanti region. There's a lot of M. ulcerans in this area. People come down to the water to wash clothes, to drink, to bathe. There's a lot of water contact. Even if there's a tube well present in the village, often people will come down to their water source and really have a lot of body immersion in this. And we found when we were working there that there were crocodiles. And we had been told this, but we didn't understand the word. And we stopped and we talked to the chief every place we go, and then we went, and then they're shouting excitedly, and this was in the water with us. So that was most of them are smaller than this, but that's been kind of interesting. So what do we know about the organism in the environment? We know that it's associated with the first work that was really interesting came out by Françoise Portals. She reported in The Lancet in 1999 that she'd picked up some insects, and in 3 different genera she had M. ulcerans DNA. And other people, this M. ulcerans DNA has been found in predaceous water insects in Cameroon, in Cote d'Ivoire, in Benin, and Togo. So I think this is a real association. The DNA is there. She's also found it in insectivorous fish. Other people have found it associated with plants. Laurent Mercier has done a lot of work in this regard. He also found it in snails. But all of this has been very small scale sampling and not the way environmental scientists do this. There's also laboratory models. Laurent has done some beautiful and elegant work showing that he has taken and collected biting predaceous necorids from swamps in Spain and infected them with particular species of Mycobacterium ulcerans, and he can get a productive infection in these insects. But when we've tried to repeat that data with African M. ulcerans and African insects, we can't get any replication. So I think you want to go from the field to the lab, but the interpretation of the data can be very tricky. So here's our big view. This is all of what we're involved in, going down from the landscape to the community. And we want to find out what all the ecological connections are, and this really amounts to kind of what's eating what. And we're trying to reconstruct these chains of organisms in the environment that can be host to M. ulcerans. And we started off with the Landsat data. Okay, so this is pretty interesting. It's satellite data. So you buy these images, and they'll show you the country that you're looking at, and they're picking up on electromagnetic energy and reflectance and absorbance, and different land covers have different signatures. However, there's a caveat. There's this thing called ground truthing, which I always sort of wonder what that was about. And just went back to do this in Ghana in October. And basically, you go to all these different sites, like a small water body, a rice field, a banana plantation, a teak forest, and you take GIS coordinates, and then you go back and you look at your map and you see, can you actually distinguish what those are? You should have 85% accuracy. We're at 75%, not too bad. So here's some data from Benin. These colors here are showing you the different landscape types, and over here, overlaid on that, on this side are all the villages in Benin, okay? And over here are all the villages that have Buruli ulcer. So one thing that comes out very clearly is that Buruli ulcer is much more common in the southern part, Buruli ulcer is much more common in the southern part of the country than the northern part. And furthermore, in the areas that are very dry, such as these areas, there are a lot of villages in these areas, but there's no Buruli ulcer. So this kind of data, we're using this kind of data, and really so far, sort of aggravating because it's confirming what everybody's known, that there's an association with water. It doesn't occur in the savanna areas. But you can go further with this, and we're trying to. We're trying to do this in Ghana now. I just wanted to show you, here is Lake Volta. These are some of our study sites. Huge amounts of water, but no Buruli ulcer there. The rivers aren't shown here, but in these sites where we do have Buruli ulcer, there are rivers running through. These are sort of drainages for these different rivers. It's a very hilly area. The biggest problem we've had so far has to do with resolution. If you really want to see what's going on at the village level, you have to have 1 to 2 meter resolution so that you can actually see like this, which you can't. It's kind of astounding. It's like Google Earth or Berkeley or something. But when you look at Ghana, by what you can find on Google Earth, you don't have this sort of resolution. And you have to pay for it. But I think about, John brought up John Snow. I mean, that basic experiment where you map cases to houses has never been done for this disease. And that's what we're hoping to accomplish with this. So what we've shown with our landscape work so far is that urban areas have less BU than rural ones, association with smaller water bodies. Human density doesn't seem to have a big effect. Agriculture, presence of agriculture is important. Forest, where you have forest, you don't seem to have an overlay of Buruli ulcer, but when you have this border between forest and agricultural land, that may be something we need to look at more carefully. And as I said, we've really been limited by low resolution. And we've worked with modelers on this guy Wagner, who has done, really did most of the statistical analysis for that. So we've been involved in the standard ecological sampling methods and the development of probes to look for the organism in the environment. Now, it seems like a trivial problem, but if you want a specific probe against an organism in the environment, you don't know what else is out there. So you can never know your probe is specific, right? But you can hedge your bets by having multiple primer sets and so on, and hoping that, statistically it will fall out, and that's the approach that we've taken. And we've looked, this is basically our strategy. We go to villages, we're going to 160 villages. I think we finished 98 so far, endemic and non-endemic. We draw a transect through a water body in a village. We lay down a quadrant, a meter quadrant, take everything out of that, pick it, separate all the insects, all the species. We filter water. We also add glass slides and collect biofilm and collect soil. bring it back to the lab, and PCR thousands of samples. Okay, so it's very brute force kind of work, and what we found is that the DNA is widely distributed in the aquatic environment, but not in the terrestrial environment. We can begin to reconstruct feeding groups, and we also found that M. ulcerans is present not only where the disease is present, but Buruli ulcer, areas where there's no Buruli ulcer, you can also find the organism in the environment. Now, that sort of naively disturbed us at first, but this is kind of the rule for an environmental pathogen, because humans are accidental. So if you look at Yersinia pestis, every single county in Arizona has infected rodents, but only a handful of counties have cases of human plague. So there are complex factors that lead to occurrence of disease, and having the organism in the environment is necessary but not sufficient. And this is shown on a map here where we have this plus. You can see that that's a situation where we have M. ulcerans and we have a Buruli ulcer case. So that's wonderful, it's very consistent. Now we have cases here where we detect M. ulcerans in the environment, but there are no cases. Now, one big problem in a rural area is underreporting. Is this because sometimes we've gone to villages that were non-endemic and seen somebody with Buruli ulcer? So that could be part of it. But probably there really are places where M. ulcerans is in the environment, but there are no cases. We also have some examples where we're not detecting M. ulcerans in the environment. So maybe this is a sampling error, but there is a case. So at this point, we're trying to sort this out. And I think one of the most interesting areas is this area here along the Volta, where we have lots of sites and where we've tried to use these sites to test our PCR probes. And basically, there is no M. ulcerans or Buruli ulcer in that region. So that was a major finding. We have a lot of hypotheses about how transmission might occur, how it might be passed through the food chain, but basically at this point it's going to take a lot more work before we can really show this. And so we wanted to look more closely at associations of particular communities. So we know that about 37% of our samples of plants in an area will be positive for M. ulcerans DNA. It's not specific to a particular plant, but the idea was maybe biofilm is forming on these plants, or maybe it has to do with particular communities of plants. So this has been addressed by using a method called non-metric dimensional scaling. And the way this works is each one of these spots has two pieces of information. One, it's a site. It's a geographical location. The second one is it represents the community of plants at a particular place. And the way these are arrayed, if you have two spots that are close together, that means that the plant communities are similar. If you have one up here and one over here, this means these two plant communities are very different. So if you look over here, we have three sites, and they're close together on this map, and so that means that the plant communities, that means the different species compositions from these three different sites are very similar. Now we want to take this data and overlay on it our data from the bacteria. Where's the bacteria in these sites, or where's the disease? Okay? So now we're taking BU case data, and there were those three sites I showed you before that showed three villages with very, very similar plant communities. And you can see that we've got both endemic and non-endemic regions in the same area. And basically, when we go through all of this data, the bottom line is that there is no association with a particular plant community and presence or absence of Buruli ulcer, the disease Buruli ulcer. So now we look at the bacteria. What about the distribution of the bacteria? Is that influenced by plant communities? And basically we get the same story. We really can't show that you're getting the red and the blue kind of distributed all over the place. So it doesn't appear that particular plant communities, and a plant community, what plants grow there is a reflection of complex soil chemistry, water chemistry, all these different factors. And none of these really can explain the presence or absence of M. ulcerans or Buruli ulcer. And we've used sort of the same sort of argument to look at insect populations. Yeah. Okay, so I'm almost done. So because there was some evidence for insects, and we go through and we do the same studies with insects, basically, we can't really find an association between Buruli ulcer or M. ulcerans and insect communities, and we do specific studies asking, can the numbers of insects here of this type and the amount that are infected with M. ulcerans explain disease? It can't. So basically, we don't think they are major vectors of disease. I just wanted to show you one example of a positive hit, just so you would see what it looked like. And this is very preliminary data, but in this case, we're collaborating with Mike Kaufman at Michigan State, and he's looking to see, is there an association between the particular members of a bacterial community at a site and the presence of M. ulcerans? And all of these sites are sites where we have found M. ulcerans in the environment, although he hasn't found it in his samples. And these are all clustering here in a discrete cluster separate from here. And so this is what positive data would look like, that maybe there is a positive association here. Maybe M. ulcerans is killing certain members of the bacterial community. Maybe the presence of other members of the bacterial community are unfavorable for ulcerans. So basically, our research priorities, we want to identify where it replicates in the environment and the effects of environmental degradation, develop small-scale disease ecology maps, and look at what cultural factors might play a role in this. In terms of diagnosis, we really need a simple point-of-source diagnostic. We wish somebody would take this up, and the development of a good delivery system for antibiotics. This is my lab. And I want to particularly mention our collaborators at Michigan State University and Noguchi Medical Institute in Ghana, where we've been doing most of our work recently. And Lydia Mosey and Heather Williamson spend about 3 months, 2 to 3 months a year in the field. So thank you very much.
I can't help but ask after hearing John's talk, another parameter you might want to add is bacteriophage.
Yeah, I thought about that too.
And I wonder, it just occurred to me, I never thought of this, when if you have a bug that can't grow at 37, I wonder if there's a prophage that pops out at 37.
Yeah, our incubator went up to 37 once, and the cultures got turbid, and that really occurred to me, but we never looked.
Okay, any questions? Other questions?
Well, I was wondering whether local temperature is a difference, because if they cannot grow a high temperature, maybe I don't know whether they have dry areas where more the temperature is higher or lower could also make a difference.
It might, because the temperatures in the drier regions are higher. The water temperature, even though the ambient temperature in southern Ghana is lower than the ambient temperature, but it's not as hot as in the northern region. That's a possibility.
So, spectacular use of Landsat data to study neglected diseases. I've never seen that before.
Yeah, you haven't gone to ecology meetings.
Amazing. The question I had was, there's a lot known about drug sensitivity and resistance now in other mycobacteria.
Yeah.
And so, because you have the genome, does it give you insights about how to tailor antibiotic treatments? It's quite shocking, I guess, to me that you started experimenting with treatments only in 2005. And so how do you pick the pharmacopoeia to apply?
Well, we did it based on TB, and the TB people are really concerned. They don't want us to use rifampicin. But the earlier study said that antibiotic treatment didn't work, and nobody readdressed the issue, until they had newer drugs. So so far the genome in that regards is not different from TB. I mean, we don't see anything obvious. We'd like to inhibit the toxin synthesis. And that would be an interesting approach. But we don't, I think the drugs that work against environmental TB usually work against all of them. They're not, the environmental mycobacteria, they're not really specific. Maybe I didn't. Okay.
Okay, so thanks, Pam.